Good afternoon, good evening. I'm Hervé Affagard. I'm the co-founder of MaaT Pharma, and I'll be the host for the discussion today. In terms of the logistic, there will be a Q&A session at the end of the meeting. You can use the questions button on the bottom of the presentation, and they will be sent to us, and we will answer your questions. So I'll start with the introduction. I'm together today with, I'm together with Professor Mohamad and also Professor Florent Malard. They are both working at the Saint-Antoine hospital. Mohamad is the head of Hematology and Cellular Therapy Department, and Professor Malard is also on the Hematology and Cellular Therapy Department of Saint-Antoine Hospital. So I'll start with the introduction of MaaT Pharma. So MaaT Pharma is today the leader in the field of hematology oncology when it comes to microbiome modalities. We have developed a platform which is called MaaT, Microbiome Ecosystem Therapies, and our niche, our market entry strategy is focusing on hematology oncology, and our objective is really to enhance patient survival. We have been very fast in terms of the execution regarding since the inception of the company back in December 2014. We are a 9 years old company. We have been created in December 2014. We have treated our first patient in 2016. We developed a technology which is leveraging on the concept of pooling, so that we can achieve a high level of standardization of the product. And this is the first FDA approved authorized pool microbiome product. We have taken 7 years to enter in the phase 3. That's the data we're going to be presenting today on the phase 3 for RS, and also more recently we have built our manufacturing facility that has been very fast, as we have taken only 12 months to complete this new manufacturing facility. In terms of the way our drugs are working, here we represent the gut of a patient, and you'll see that the blue layer is the gut microbiome, and as you know, 80% of the cellular host defense, they reside within the gut. And what our drug would do is that we will reset the blue layers. So what we mean by resetting is really about increasing the diversity of the microbiome, because we have evidences showing that you need to be diverse to get a good clinical response to the for the patients. And our drugs will be targeting this blue layer, and the mode of action basically it's to restore the microbiome microbial homeostasis and network, so that it will increase diversity, eradicate the bad bugs, and in turn it will also restore the immune homeostasis. That's the way we're gonna be improving overall survival in patients. Focusing in HSCT on the left-hand side, you see that that's an illustration of the benefits of the diversity. You see that the highest the diversity in patients, the highest the survival. If we focus on the left graphic, you see that you can improve by like 50% the survival of the patient if you are diverse at the time of the engraftment of neutrophil. So this is a pretty significant evidence that we are leveraging on. The different products that we are developing, that's two range of products, MaaT-N and MaaT-C, and that's because they are native products. They come from donors. C, it's because they are co-cultivated products. We don't rely on donors. Today, we will be focusing on the MaaT-N products, which are MaaT13 and MaaT33, which are the products that we are evaluating into the clinics in the hematology oncology setting. We use our data science platform to better understand the mode of action, stratify patients, but also develop the different products. In terms of the pipeline, we have 2 products, 4 different clinical trials. 2 are on the field of hematology oncology, that's ARES and Phoebus, that we will be discussing today. And we also are expanding in solid tumors with metastatic melanoma patients, where we use MaaT in combination of immunotherapies. And we have also launched recently a proof of concept in neurodegenerative disease. With that introduction, I will now let Professor Mohamad Mohty introduce the work that we are doing with MaaT in GVHD.
Thank you very much, Mr. Affagard, and good evening, good morning, good afternoon, everybody, wherever you are. It's a great pleasure to be with you, and I've been asked for the next few minutes or so to give you a brief summary about the results we have generated from the early access program using one of the MaaT assets, namely MaaT that was mentioned by Hervé. And this is in the setting of refractory GI acute graft versus host disease. So let me start first by highlighting what is this product? What is MaaT? Actually, it is a maximum density product which would allow for fast engraftment of the microbiota in acute situations. And this is definitely what we need when we are facing a life-threatening condition like acute GVHD. So in contrast to single donor homemade products, this is a true drug made from a pooled microbiota with high richness, very well characterized, high diversity, a sort of a full microbiome ecosystem treatment containing exactly the beneficial effect of the Butycore. And it is administered in the form of enema, and I'll show you here in the studies we are performing. Usually we would go for 3 doses. And the current indication is actually gastrointestinal acute severe graft versus host disease. And our initial results were published as part of a seminal phase 2 trial called Heracles, which included 24 patients, and this was published a few months ago in eClinical Medicine, a very high impact factor journal from the Lancet series. And today, what I'm going to present to you are the results of 111 highly advanced patients with GI acute GVHD who actually received the MaaT product as part of an early access program in France and across Europe and the world. But these are 111 patients with mature and well characterized data. But I must confess that we have already more than 200 patients being treated today. So for those who are not really familiar yet with acute GVHD, actually this is one of the most serious complications of allogenic stem cell transplantation. And allogenic stem cell transplantation is the only curative treatment option for many patients with leukemias, especially myeloid leukemias, because despite the advent of novel therapies, cellular therapies, unfortunately, the outcome of these patients is very dismal. And unfortunately, one of the severe and key complications of allogenic stem cell transplantation is the development of acute graft versus host disease. And this could affect the skin, the liver, but also the gut. And actually, the gut involvement is usually the most life-threatening, I would say, condition. And that gives you around 22,000 patients per year receiving transplant and you end up actually with around 10,000 patients who are candidate to some curative treatment, and out of them, 1/3 of them are going to be steroid, but also ruxolitinib resistant and refractory. But I'll come back to this in a few seconds. But the bottom line, why MaaT is a key drug for treating these patients with very dismal outcome, it is because we have very strong body of data highlighting that the loss of the microbiota diversity, in another word, what we call the dysbiosis, is a key initiator and a trigger for these severe cases of acute GVHD. And this is favored by the chemotherapy, the irradiation, but also the multiple courses of antibiotics that these patients are receiving. And once they develop acute GVHD, you usually treat them with steroid, corticosteroid. This is the best we can do since now more than 40 years. But unfortunately, not all of them will respond to steroids, but also some patients may not tolerate steroid. And until recently, the outcome was very dismal. The mortality was over 80%. So you would imagine a terrible situation. Recently, we had ruxolitinib, a JAK2 inhibitor, which has been approved for those patients in steroid refractory acute GVHD. However, one should bear in mind that unfortunately, not all patients will respond or are able to receive ruxolitinib. And most importantly, some patients are even resistant to ruxolitinib. And in this situation, there is a clear unmet medical need. And this is where comes the MaaT use and story. And this is about this early access program that is still ongoing today in Europe. And I mentioned more than 200 patients were already treated. So this is about patients who are steroid refractory or even steroid dependent with a very severe gut acute GVHD. Usually it goes from grade 2 to 4, but most of them are grades 3 and 4. And actually, this is used as a monotherapy or even in combination, because as you may guess, in an early access program, it's not a prospective controlled trial. It's just according to the attending physician decision. And obviously, these are patients who are not eligible to our ongoing registration pivotal trial, phase 3 trial called ARIS. And usually, the administration would require a few hours of discontinuation of systemic antibiotics. You can see here the characteristics of the patient. The median age is 57, but you can give it, with no age limit. The older patient was age 74. And actually, these were really highly advanced patients who received a median of 3 lines of therapies for their acute GVHD. So I can assure you, these are patients who are, in theory, supposed to be in supportive or palliative care. And you can appreciate that the majority, 85%, were steroid refractory. And as I mentioned, the majority, 91%, had the most severe, I would say, advanced cases of acute graft versus host disease. So these patients received the MaaT drug as an enema, and the results are quite astonishing. You can see here on the left-hand side, the GI overall response rate of 53%. But what is really even more important than these 53% is that the majority of the patients are going to be in complete remission or even very good partial remission. And we know that this is a key and mandatory step to improve the survival and the outcome of these patients. And actually, this is what you can see on the right-hand side of the slide, where those patients who are responding to the MaaT therapy, are actually enjoying a 67% probability of survival at 1 year. And this has never been described in the past. And this is highlighting the huge benefit of this life-saving therapy. And if you focus actually on the most advanced patient, which means these are the patients who are not even only steroid refractory, but also ruxolitinib refractory, because ruxolitinib is an approved and widely used drug now in acute GVHD. And these are patients similar to our ongoing phase 3 registration trial. And the results are even more amazing, I would say, because you have a 61% GI response rate, including almost exclusively complete remission. And this will translate into an incredible, really astonishing overall survival rate and responders, 81% at 1 year. And please don't forget, I mentioned to you that the mortality in these patients at 2 months or 3 months would be more than 80%. Now we have a probability of survival of more than 80%. And you can achieve these amazing results actually with an excellent safety profile. 16% of the patient reported an adverse event which was probably related to the product, but actually there were no pathogen transmission, because that was some concern initially, because these patients, they do have a fragile gut, and there were no deaths attributed to the drug itself. And currently 55 patients are alive at last follow-up. So one would reasonably, I would say, conclude and with strong confidence that MaaT is a safe, effective drug for steroid, but also ruxolitinib refractory acute GVHD, given the very high response rate and the positive impact on survival. And that has set actually the stage, although the early access program is ongoing, to run this phase 3 clinical trial, which aims to include 75 patients across Europe. So with this, I will stop here. I think we'll take questions later, but I'll hand it over again to Mr. Affagard.
Thank you very much, Professor Mohty. So just to remind, I see there's already questions, but for the benefit of the audience, you can ask your question as we speak. No problem. We will be dealing with the questions at the end. So now we have finished with the presentation of the early access data with MaaT. Now we're going to be going to the next product, MaaT33, and that will be with a presentation of Professor Florent Malard. Thank you very much, Professor Malard, if you want to continue the presentation.
So thank you, Mr. Affagard. And we are now going to discuss the FIBRUS trial. So this is a multi-center randomized double-blind phase 2b trial that evaluates MaaT33 to improve the outcome in patients receiving allogeneic stem cell transplantations. So I am going to present you what is the rationale to develop this MaaT33 in allogeneic stem cell transplantations. We know that in patients that are going to undergo allogeneic stem cell transplantations, they have at baseline a healthy microbiome, but before we start allogeneic stem cell transplantations, they receive some high-dose chemotherapy, some antibiotics that will induce some dysbiosis because of the treatment, in particular for the treatment of acute myeloid leukemia. And when we are going to perform the allogeneic stem cell transplantations, again, they receive high-dose chemotherapy. This is part of the transplantation platform. They need to receive this high-dose chemotherapy for disease control and engraftment for the success of allogeneic stem cell transplantations. And almost all patients also will have to receive some antibiotics with a broad spectrum, so it will have an important impact on patients' microbiome, and it will induce an important dysbiosis. And in fact, over the last decades, a lot of studies clearly demonstrate that this dysbiosis have an important impact on patients' outcome. It will, of course, induce a higher risk of infectious complications. This is easy to understand. We have some dysbiosis, we have more bad bug, and it will lead, of course, to more infections. But we have also some data that show that it can increase the risk of relapse of the underlying disease because it will interplay with the immune systems, and probably it will lead to a decrease or a decreased efficacy of the allogeneic stem cell transplantation on an increased risk of relapse of the leukemia. And as Professor Mohty just told you, this microbiome is closely related to this graft-versus-host disease, to these life-threatening complications, and in patients with this low bacterial diversity, it will increase the risk of graft-versus-host disease and increase the risk of graft-versus-host disease mortality. And this dysbiosis is also associated with the delayed adaptive immune recovery that will also contribute to the increased risk of very severe infection after stem cell transplantations, and that will overall lead to reduced overall survival. And in fact, this reduced overall survival was confirmed in a very big study that included more than 1,000 patients that were not treated in a single centers, but it was some patients treated in the US, in Europe, in Japan. So it was some patients treated all over the world. And with more than 1,000 patients, they were able to demonstrate that patients that have a higher diversity at engraftment after stem cell transplantations have a significantly higher overall survival. So it was a very powerful finding, because we are speaking about patients with acute myeloid leukemia, very severe disease, and in fact, this gut microbiome, this dysbiosis, was a very strong predictor of patients' survivals. So, based on these findings, we need to develop an interventions to restore this gut microbiome dysbiosis, to prevent the dysbiosis and to improve the patient's outcome. And this lead to the development of this MaaT 33. So this is similarly to the MaaT 13. This is a pool microbiome with a high richness, a high diversity, full ecosystems that which contain a high number of butyrate producing bacteria, so this so-called butycore. So these butyrate producing bacteria, some bacteria with some immunoregulatory properties that are been shown to be protective and to be associated with a good outcome after allogeneic stem cell transplantations. And what is very interesting with this MaaT 33 is that it can be given as an oral product, and this is a lyophilized capsule, so the patients are able to take their capsule at home, and this is an home delivery, very easy. So what is the treatment schedule planned in this phase 2b clinical trials? It will be a first treatment for 1 week before we start the conditioning regimen, so before we start the allogeneic stem cell transplantation. So they are planned to receive 3 capsules per day for 1 week, and we will stop day 7 before allogeneic stem cell transplantation, because this is the time where we are going to start the chemotherapy. And after neutrophil recovery, so this is after allogeneic stem cell transplantations, we'll again give 3 capsules per day. So it will be around day 18 after allogeneic stem cell transplantations, and we'll pursue the treatment for up to 90 days after allogeneic stem cell transplantation. So this is a very long-term treatment aiming to have a full microbiome diversity recovery to improve the patient's outcome and prevent allogeneic stem cell transplantations complications. So these dose of 3 capsules per day were based on the phase 1b Simon study that was conducted in 6 centers in France to establish what was the right doses of MaaT 33 to have an engraftment and to restore gut microbiota diversity. And in fact, we use some patients that have a diagnosis of acute myeloid leukemia that receive high doses chemotherapy, a lot of antibiotics that induce some important dysbiosis. And when patients recovered, when we stopped all the antibiotics, they were included in these phase 1b clinical trials. And we have several cohorts. In the first cohort, we give only one pills of MaaT 33 weekly for 2 weeks, so 2 pills. In the cohort 2, it was 1 per day for 1 week. Cohort 3, 3 per day for 1 week. And in the last cohort, it was 3 per day for 2 weeks. So it was this dose escalations clinical trial that is typical for the phase 1 clinical trials. And in fact, as you can see here, this is baseline. So this is when we are going to give the first MaaT 33 treatments. This is 1 week after, 2 weeks after. And here, this is after patients receive further chemotherapy that are planned for the treatment of acute myeloid leukemia. And as you can see here, in all cohorts, we see a significant increase into patients' microbiome richness. So that is to say that we find more bacteria 1 week after the treatments, and it was further increased 2 weeks after in most cohorts. It was only in the first cohort when we give only 1 pill per week that we don't have this strong persistent into the increased richness, but we have a very low doses of treatment in this case. And when we look at the engraftment, we are also able to see that in all cohorts we have a strong engraftment. That is to say that we find the bacteria that are within the MaaT 33 in the stool of the patient. So we have an increase into the richness of the product that were associated with the good engraftment of the product, including this butyrate, so this beneficial butyrate producing bacteria. So overall, we find a robust and persistent engraftments. We see that the engraftment follow MaaT 33 treatments that correlate with an increased anti-inflammatory markers. And we have a very good safety profile since we have 22 patients exposed, 22 that completed and that received the entire treatments. It was a very good compliance because it was some small pills that was easy to take for the patient. So all patients takes all the pills, and we have some very positive DSMB meeting regarding the safety of the treatment, which each time go. So based on these phase 1b clinical trials, we're able to design this phase 2b randomized Phoebus clinical trials, and we have a very good primary objective since we aim to improve the overall survival in patients at 1 year after randomizations. We have some very important secondary endpoint, of course, regarding the safety of the product, of course, also the GVHD-free survival at 12 months. So we expect also to reduce the incidence of graft-versus-host disease with MaaT 33. We'll, of course, look at the incidence of grade 2 to 4 and grade 3 to 4 acute graft-versus-host disease and of chronic graft-versus-host disease. That is another kind of graft-versus-host disease that we can see after allogeneic stem cell transplantations. Overall, we also want to evaluate non-relapse mortality, infectious-related mortality, graft-versus-host disease-related mortality, and also the survival without relapse and without graft-versus-host disease. We also want to assess infectious event. It will be part of the safety analysis also on patients' quality of life. And finally, we'll of course assess microbiome compositions to evaluate engraftment of the product and the increase in richness that is expected in patients that receives a MaaT 33. So here is a study flow chart. So we plan to include almost 400 patients, 397 patients in these international studies. So patients will be screened. Of course, it will be the first screening visit and inclusion visit that will be between day -21 and day -14 before the day 0 of the allogeneic stem cell transplantations. They then will be able to receive the treatment for 7 days, and it will stop at day -7 to to start the chemotherapy for 1 week before patient will receive allogeneic stem cell transplantations. After conditioning regimen, there is a decrease into the neutrophil that is expected at engraftment. So when neutrophil go up, we'll be able to start the second treatment period. So from neutrophil recovery up to 3 months after stem cell transplantations, and we'll have some follow-up visit at 4 months, 6 months, 9 months, and 12 months when we will assess the overall survival. That is a primary endpoint. We'll have a stratification to be sure that the groups are balanced. Of course, we'll stratify based on the disease, underlying disease severity, this using the disease risk index that is an internationally validated stratifications parameters to assess the risk of the different kind of acute leukemia that are going to receive transplants. And we'll also stratify the patients based on the HLA compatibility. Within 1 month, some patient that will have some HLA identical donors, and in the other on the patient with some kind of HLA incompatibility. So where we have some HLA mismatch, just to be sure that the groups will be perfectly well balanced. So regarding the inclusion criteria, we decided to focus on patients age over 50 years because this is where we really have an unmet need to improve patients' outcome. But of course, if it works in patients over 50 years, of course, it will work in younger patients with an hematologic malignancy that need to receive stem cell transplantation, of course, with a neutrophil level above 0.5 because we don't want to give the treatment in patients that have very low neutrophil level per regulatory guidelines. And of course, we will only include some patients that receive some broad spectrum antibiotics within the last 90 days prior to inclusion, because we want to be sure that we are going to treat some patients that already have some dysbiosis, where we will have a true benefit from the use of MaaT33. We will on the opposite, just exclude a patient that receive antibiotics at time of the inclusion, because otherwise we'll have directly a detrimental effect on the treatments. And we'll also be sure that the patient have a good general condition, that we have a donor, that we can perform the stem cell transplantations. But this is some very classic inclusion criteria for a protocol related to stem cell transplantations. Regarding the inclusion criteria, we decided to exclude some patients with non-myeloablative conditioning regimens, because this is some patients that almost no have any neutropenic phase, did not receive antibiotics with very low dysbiosis, so there will be no benefit to give MaaT33 in those patients. And also the patient that receive some very high, very intensive myeloablative conditioning regimens with high dose TBI, with high dose busulfan combined with cyclophosphamide, because this is some conditioning regimen that are almost everybody stopped using because of the toxicity, and we think that the toxicity of these conditioning regimens will be too high for our patients. We also exclude several kind of patients regarding different graft-versus-host disease prophylaxis with alemtuzumab, vedolizumab, abatacept for the purpose of the homogeneity of the cohort. And of course, some patients that have some contraindication to allogeneic stem cell transplantation will be excluded, but anyways, they will not receive stem cell transplantation, so they should not be included. So what is the study status? So I am very happy to tell you that the first patient were included on dose in November 2023. And so far, we have France and Germany that have active countries with some inclusion. And the next step will be to open United Kingdom, Spain, Belgium, and Netherlands. So now I will give up again to Mr. Affagard for the conclusion. So please.
Thank you very much to you too. We are lucky to have two true pioneer on the field of microbiome and hematology. So thank you very much for sharing your experience on this field. So I'll go to the conclusion. We have a lot of questions, so I'll try to be fast. In a nutshell, we have two different sequences. The first one is MaaT 13. So the first step for us would be to complete the European part of RS with MaaT 13. We expect to have enrolled all the patients and then get the primary endpoint, because as you have understood, this is day 28 after the treatment of the patient that we evaluate the primary endpoint. So it's pretty short. So we expect to have by mid-24 the primary endpoint. Because it's a first-in-class product, we will also follow the patients during a year, even if now with the early access data, we know that we have a pretty significant safety database. But as per protocol, we will follow the patient during a year, and that will be the end of the clinical trial by mid-25. From that, we will then, if the results are good enough, which is what we hope, we will then file for a marketing authorization in Europe. For MaaT 33, it's a longer clinical trial. That's in total 387 patients. We expect to have in the second part of 24 the first DSMB, which will be taking place. Then we will continue the enrollment of the patient. In 25, we're gonna have the second safety interim analysis, safety analysis, and the results will be known by 26. Here again, we're going to be following the patient during a year after the treatment of the last patient. Coming back to the initial slide, MaaT, microbiome ecosystem therapies. We are leading the field in hematology oncology. We have demonstrated as part of the phase 2 in Heracles that we can modulate the immune activity in a very severe conditions such as GVHD. Now we are pursuing the phase 3 with RS, with the endpoint which will be mid-24. So we are in a good direction with that, and we have accumulated very strong data. I would say that's the most important, and now we are expanding with the capsule, which allows also for ambulatory treatments, and that's also ongoing. Focusing on the EAP, it has also helped the company to generate revenues. They are all revenues from France, which is in Europe the only country where it's feasible to generate revenues. So we have been generating close to 2 million euros as of September, and that continues on the fourth quarter. We expect to have a very good year in terms of demand. Our cash position is close to 32 million euros as of end of September, and that is taking us to the second quarter of 24 as we speak today. So there's a lot of questions. We're on time. We have 20 minutes, and I'll take your questions. Name the person who asked you the question, and I'll assign one of the speakers to the answer. The first two questions would be for you, Professor Mohty. One is from Dr. Rimbaud. The other one is from Jacob from KBC. The first question is, do your results of microbiotherapy in marrow transplantation allow us to consider it in all iatrogenic alterations of the intestine in oncology? And the second question is that based on the data set that you presented today, 111 patients, do you think this data set could be a benchmark for what we could expect in the phase 3?
Yeah, no, excellent questions. Thank you very much. And they are absolutely relevant and timely. So the first question about whether we can extrapolate the benefit of MaaT 13 and MaaT 33 eventually for other disease conditions, the the short answer is yes. Actually, in every single disease condition where you would expect dysbiosis because of antibiotics, because of chemotherapy, radiotherapy, any condition leading to dysbiosis actually would benefit. And for instance, we have already some evidence in the literature about dysbiosis in the setting of CAR T cells, in the setting of bispecific antibodies, but in the solid tumor and oncology in general. So actually, this question sounds like music to my ears. And actually, I believe today we have sufficient evidence to consider that the modulation of the microbiota with products that, like the one manufactured by MaaT, actually can be an excellent partner for any treatment in cancer, I believe, and probably beyond. But this, I'm more in the cancer field, I would say. The second question is about what can we glean, I would say, from the current early access program results, and whether this can give some good indication for the ongoing ARIS trial. Actually, these results are really excellent. So in theory, it gives you a flavor on what to expect from the ARIS phase 3 trial. Obviously, we don't know what are the results of the phase 3 trial yet, although the latest DSMB and the press release that followed from MaaT suggested that the results are excellent and even beyond expectations, because obviously in the ARIS trial, the population is very homogeneous, in contrast to the early access program where you cannot control. The inclusion criteria are clearly about those patients who are ruxolitinib and steroid refractory according to the official and I would say universally admitted definition. So in summary, I think the early access program results are already excellent, and I'm extremely positive about the rest of the story.
So the third question is from Jacob. It's related to a potential baseline characteristic of the patient so that we can predict who gonna respond to the treatment or not. What we have seen in the phase 2 is that the more dysbiotic you are, the better. And it makes sense. It was not obvious to us, but at the end of the day, it makes sense, because the more you have the ecological niche, the better it is for the patient, for the product to engraft. So what we have decided with the scientific advisory board is for the phase 3, to add a pre-treatment of vancomycin for 2 days so that we can break the ecology for all patients. So that's another way to answer the question. So yes, the more dysbiotic you are, the better it is. But it's not that we are taking only those with a dysbiotic microbiome. We are also making an average microbiome for all patients, and by giving antibiotics to all the patients. We need to free up the niche for the patient to be the most efficient. The question is still valid when it will not be iatrogenic dysbiosis, but I would say more functional dysbiosis, where you have a limited dysbiosis. That would be a different question, and here the biomarker question will make sense. But here in the field of hematology oncology, that's the choice we made to make sure that the ecological niche is freed up, and that's improved the level of response. Then we have a question from Christophe Dombu from Kepler. That's for you, Professor Mohty. That relates to the fact that we are selecting the patients based on their GI status. And the question is, would your treatment be efficient also beyond GI patients?
Well, obviously, again, this is an interesting and important question. Based on the mechanism of action, our first target is the GI, of course, and especially that in acute GVHD, the life-threatening organ is actually the gut. Having said this, what we have noticed, at least empirically from the early access program, some of the patients, is that the other organs are also responding. So probably there is a sort of a systemic effect and a clear benefit irrespective of the organ, but definitely the target organ based on the mechanism of action of the drug is the GI.
Thank you. The next question is from Jacob, KBC. Can you share any updates regarding the status of your interaction with the FDA with regards to initiated clinical activities in the US, and what are the timelines? So today there's nothing preventing us to open centers in the US. We have been discussing last week at ASH San Diego with a lot of investigators, and they are really willing to help us with the expansion in the US. So the IND is cleared. As you know, we have been on hold with the FDA during a certain period of time. We get out from the hold. When we get out from the hold, it's because also we have done some modifications on the CMC process. Those modifications have been made, the products have been produced, so there's nothing preventing us to continue the clinical trial, to expand the clinical trial in 24. The timelines will also depend on the financing of the company, of course, but there's nothing preventing us to expand in the US. The IND is cleared, the centers have been identified, the products are here, the CRO has been selected, so there's nothing preventing us to expand in the US, but also we have been focusing in Europe, and we have the capacity in Europe to recruit all the patients we need, but of course we would like to go to the US, so that will be an activity for 24. So then the next question will be for you, Professor Mohty. What was the main reason why RS-like patients were not eligible for RS trial in the EAP?
Yeah, very good question. Well, first of all, and the most frequent one was about those patients who were treated in centers who were not participating to the ARIS trial. And obviously, given the brilliant results that we already had, it was ethically mandatory, almost I would say, to make such an early access program available for patients everywhere in France and across Europe and even beyond Europe. Also, there were patients who did not fulfill some of the criteria of ARIS, because ARIS is really a very well-defined population of steroid and ruxo refractory patient. But most of the time it was about the participating centers.
Another question for you, Professor Mohty. What was the feedback from the medical community at the ASH conference regarding the data you presented?
Well, I would say the feedback was extremely positive, and Professor Malard can confirm this because he was also attending. I think the community was extremely enthusiastic because these are unprecedented results we see in such a life-threatening condition. Nobody ever saw such a high response of CR, but such a high level of survival. Obviously, it's not randomized data, it's an early access program, but these results were very, very well received by the community. I don't know, Florent, whether you would like to comment on this.
Yes, I completely agree with you, Professor Mohty. It was very well received. We had a lot of questions. It raised a lot of interest, and the centers, including in the US, are very interesting to have access to these drugs. We also have some people, for example, from Australia, that say can we participate in the clinical trial? Can we have access to the product through an early access program? So for all over the world, we have this strong interest, and people are really excited about the results. So it seems very promising.
I can even add one example I experienced myself. Immediately the day after the presentation of the data, I received a case by email from the Netherlands, for instance, for a patient, a young lady struggling with the GI, acute GVHD since several months, asking desperately to take advantage and get access to MaaT 13. It gives you just a snapshot of the enthusiasm of the community.
Thank you. We have two questions on MaaT 33. The first, so that will be for you, Professor Malard. The first one is from Jacob. Can you please shed some light on the rationale behind choosing overall survival as the main endpoint for the Phoebus trial as opposed to a GVHD-related endpoint?
So yeah, thank you for this question. And this is an important question, of course, because this is always an important question when we're going to choose the primary endpoint in the clinical trials. And in fact, it was based on all the data available. First of all, we have this very important multi-center study that established that this gut microbiome dysbiosis was associated with overall survival, and not only with some GVHD-related endpoint, not only with the incidence of GI graft-versus-host disease or with graft-versus-host disease mortality, for example. So this is a first answer to this question. The other thing is that beyond graft-versus-host disease, we know that the microbiome is linked with all complications we can see after stem cell transplantation, including the risk of relapse of the underlying disease and the risk of infectious complications. So really have the feeling that using MaaT 33, it will improve, decrease the risk of graft-versus-host disease, but also decrease the risk of infectious complications, decrease the risk of relapse, and at the end have a very good effect, not only in some graft-versus-host disease related endpoint, but with the overall survival. And this is why we decided to choose overall survival as a primary endpoint.
So second question, thank you. Second question on MaaT 33, despite the 100% compliance in the phase 1, you could expect some non-adherence during the 90-day post-HSCT. In your view, is there a risk of a lowering of your study power?
So yeah, this is also an important question because it raises really which dose of treatment patients are going to take during the course of the treatment, or if it will have an impact on the, of course, of the feasibility to achieve the endpoint if we have an underpowered study. So several answers. So first thing is that we have two sequence of treatments. A first sequence of treatment that will be very short, of only 7 days before we start the conditioning regimens. And for this phase of 7 days, we'll have a very good compliance because this is a very short period of treatments. Regarding the compliance, regarding the longer phase of treatment for day around 18 to day 90, of course, there will be a few patients that may not take the entire treatment. But we must highlight that where we're speaking about allogeneic stem cell transplantations, compliance is a key issue because patients need to take their immunosuppressive drugs, need to take some prophylaxis. And when we decide to perform an allogeneic stem cell transplantation to one patient, this is something we take into account. Or, for example, we have a patient 2 months ago with a very bad compliance, and we decided not to perform allogeneic stem cell transplantation because of that, because there was two important risk of disease complications after stem cell transplantation if you don't take the immunosuppressive drug. So patients are already selected to have very compliance. So this is the first answer. The second answer to the question is that we plan a long period, treatment periods from day 18 to day 90, and we can expect that already maybe the first 15 days will be very important to restore the gut microbiome dysbiosis and will lead already to a beneficial effect. And we decided to give the treatment for a long period of time because we know that patient can have some more event, maybe have some new infections, receive some antibiotics. So this is the reason why we plan for these long treatments. But we know that already if the patients take a part of the dose, we already expect that it will be enough to achieve the endpoint of the study.
Thank you very much. There was one question, and I don't know who to assign. It's assuming you have competing data with MaaT33, do you think you could switch from MaaT13 to MaaT33? So I don't know who's going to answer that one. Could be both.
I can start. In theory, I would say yes, because the starting material is the same. However, we need to bear in mind that severe refractory acute GVHD is an acute, I would say, life-threatening condition where you would like to achieve almost a rapid, immediate, I would say, instantly correction and recovery of the dysbiosis. And if you just consider a dosage issue, obviously the MaaT13 product is more adapted. Having said this, it doesn't mean that we cannot envision that MaaT33 would become a sort of a maintenance treatment. And that is exactly the concept Professor Malard was explaining about why we took overall survival in the Phoebus trial, because actually we do expect a benefit on different outcomes. And just to add on this, because the question was about compliance, actually the results in Phoebus will be analyzed on an intention to treat. So there's no risk. But I don't know, Florent, if you would like to add something.
No, I completely agree. I think we need these high doses of microbiome. So we need this MaaT13, at least for the first dose of treatment with this GI acute graft-versus-host disease. But of course, we can use this kind of maintenance of MaaT33. And so far, we are using 3 doses of MaaT13. So maybe we'll be able to do only 1 dose of MaaT13 and after switch to MaaT33 to have this maintenance. As when we are going to treat, for example, for CMV, we have this induction treatment after this maintenance. So I think we'll have the same kind of models with the combination of some very induction with high doses of MaaT13 combined with the MaaT33 and that it will further improve the result. I really expect that.
And question on MaaT13 from a partner. What would be the historical data you would have expected if you compare to the population you had in the phase 2?
Yeah, another important question. And obviously, as you may guess, given the dismal outcome of this condition, we cannot ethically run randomized trials. So we have to benchmark with historical data. And actually, compared to historical data, the figures are almost like 180 degrees different, because with historical data in steroid refractory acute GVHD, the survival would be less than 10% at 6 months. It's probably worse if you consider the both steroid and ruxolitinib refractory patient. And now I'm showing you in the responding patient more than 70 or 80% overall survival. So although this is not randomized and a controlled phase 3, but still I think the difference is huge between what we see in we used to see in historical cases and routine practice, and now when patients are able to access such innovation like MaaT13.
Thank you very much. There's only one question we have not answered, so I'll follow up with the person who asked the question. However, we have we are at a time limit, so I'll conclude. We want to respect your time, of course. Thank you very much. We will continue to communicate anytime we have new data and new developments. So thank you very much for following the company. Thank you for your interest. And thank you to our 2 pioneers with us today. Thank you very much. And talk to you soon. Bye-bye. And happy Christmas and a happy New Year.
Merry Christmas. Happy holidays.
Thank you. Happy Christmas.
Thank you.